Today we are going to discuss about the concept crossing over. In previous lecture we are studying a linkage. Linkage and crossing over are interrelated concepts. In eukaryotes, crossing over and genetic linkage are two different but related events. Crossing over is the phenomenon where the genetic material get exchanged in germ cells. Crossing over is the exchange of genetic material between non-sister chromatids of homologous chromosomes during meiosis, which results in new allelic combinations in the daughter cells. Means the cell division takes place. It is seen in body cells as well as sex cells. Body cells called as uh, the mitotic cell division, and meiosis which occurs in a ovaries or the testes, meiotic haploid division. This crossing over phenomenon means exchange of broken piece. In simple term, exchange of broken piece of chromosome. But which chromosome? This exchange must be a in between non-sister chromatids, because sister chromatids are the Xerox copies of each other. Such a type of crossing over do not show any change, but when it is in between non-sister chromatids, at that time it matters. It develops a new combination. You can understand the sister chromatids and non-sister chromatids with help of this diagram. The sister chromatids are identical copies of the same chromosome formed by DNA replication, which is seen in this diagram. After replication, the having a similar color copies of the chromosome chromatids are formed from that homologous chromosome. Then these are occurring during cell division. They are separated from each other, and each daughter cell receives one copy of chromosome. In this diagram, after mitosis, the chromatids which are get separated, all the four are arranged in this line. You can observe blue colored two are the sister chromatids, but the one blue and one pink, that pair is called as non-sister chromatid. These non-sister chromatids show a crossing over. While sister chromatids are exact copies of each other, non-sister chromatids come from the homologous chromosomes. They code for the same gene but are not genetically identical. Occasionally, genetic material is exchanged between non-sister chromatids during meiosis, allowing for a new arrangement of genes to be passed to progeny. This interchanging, in a scientific term, a crossing over. It shows that mechanism of crossing over shows a 4 steps. It occurs in early prophase, Pachitin stage of meiotic division. That stages are synapsis, duplication, proper crossing over, and terminalization. We see one by one. Synapsis, also called as pairing or bivalent stage. The homologous chromosome present in germinal cell come closer and pair longitudinally. This process is called as pairing or synapsis. Takes place during the zygotic stage of the prophase first. The paired chromosomes are called as bivalent. Synapsis, which begins during the zygote, continues up to the pachytail. The force of attraction is called as synaptic force. Then the duplication of chromosome. The bivalent undergoes duplication during pachytail. Each chromosome now consists of two chromatids, but the centromere will not divide. Thus, there will be four chromatids tetrad stage. And in this tetrad stage, the possibility of crossing over is there. This takes place at the tetrad or the 4-strand stage. In this diagram, this is tetrad condition and this is chiasma formation or actual crossing over beginning. In this middle diagram, you can observe that pink chromosome and blue chromosome shows a crossing over. Cross simply crosses each other. Due to that adjacent point, a point is formed called as chiasmata or chiasma. This chiasma is most important due to that chiasma formation, two segments get exchanged after breaking at that point. This takes place at tetrad or the 4-strand stage. At the time of crossover, two opposing non-sister chromatids have a break at identical points. This is brought about by action of enzyme endonuclease. The two chromatids exchange an identical length of the genome. After exchange, the segments fuse the chromatid due to action of enzyme called ligase. This crossing over is physically demonstrated in the form of chiasma. In this diagram, you can observe the both tips of this diagram shows the exchange. In this way, these two segments are changed as well as these two tips are also changed. Hence, these are called as recombinant chromatids. Next is a terminalization. After exchange of segments, the two chromosomes start moving away from each other as synaptic force lapses. The separation begins from the centromere and moves towards the ends of chromosome. It is uncoupling of chiasma, that is called as terminalization. During dikinesis, the homologous chromosome gets separate except at their ends. The mechanism serially can be understood with this diagram. First tetrad condition is there. Then next, that is chiasma formation at this stage. This is chiasma stage. Then crossing over, exchange of the broken segments takes place, and due to which these varieties of gametes are formed. This gamete, first blue colored, is parental, and this one also. But these central two are the recombinant. This is a one slide which shows a cell division. In this, many cells are there and movement of chromatids, the telophase or terminalization can be observed just for your sake of convenience. Next part is cytological evidence of crossing over. The crossing over between homologous chromosomes can be physically and visually demonstrated when the chromosomes have special physical parameters. Means the chromosome should have a different physical shape, then and then only evidence can be studied. Hence, the scientist Stern in 1937 has demonstrated very clearly in Drosophila, where the two members of homologous pairs can be identified as a result of translocation. A translocated chromosome has a modified configuration and can be identified visually. In one of the strain of Drosophila, a portion of Y chromosome has been attached to X chromosome, resulting in a L-shaped chromosome. While the normal X chromosome is rod-shaped, that is, physiology of both, shape of both is different. In another strain, the X chromosome is broken into two parts. On the broken chromosome, he placed a recessive eye color gene, carnation. It is denoted as small letters CAR, and the dominant eye-shaped gene bar, it is denoted as a capital B. The unbroken X with the translocated Y piece had a wild alleles plus of the two genes. He crossed this female with such a two abnormal X chromosomes with a male with X and Y chromosome having a recessive gene CAR and wild allele of capital B. The progeny of this cross showed a 4 types of female individuals, out of which 50% were non-crossover and remaining 50% were crossover types. Another 4 kinds of male flies will be produced due to fertilization by Y-carrying male gametes. Thus, of the 4 types of individuals, the first two belongs to non-crossover category, while the later two are of crossover category. Such a cytological observations suggested that an actual exchange of chromosome segment takes place. This can be understood with help of this diagram. The physiology change of chromosome can be observed with help of this diagram. This is explaining the Stern's experiment. In this first, female Drosophila have the gene on one X chromosome which is broken, diagrammatically shown in this first chromosome, broken piece having a genes CAR and capital B. Another X which has a plus genes, but it has a Y piece which is attached to that, and because the attachment of Y piece, it becomes a L shaped. Then in meiosis it develops such a type of, after tetrad condition, 4 types of gametes, which are, out of which first two are parental type and second two means these are showing a single crossover and due to which a single gene is replaced, that is piece of Y is exchanged with a that end is exchanged with this end having a gene CAR, and due to such a type of two genes which are similar to parent and two are having a crossover, they show the possible combination. 8 combinations are there. This male has these two, that is one X has a gene CAR and plus, and another which is Y chromosome. In this diagram you can observe the two types of progeny, that is 50% progeny show a parental type and 50% show a recombinant variety. Means cytological evidence is there due to this experiment, and you can or all the scientists can suggest or the postulate the concept which is the chromosomes has a linear arrangement of gene, as well as gene is not a another piece any material on chromosome, but it is a segment of chromosome. These are the significances. We can see one by one. The intraspecific variation among individuals is due to crossing over, and crossing over produces new grouping of genes and thus produce a new phenotypic traits. Hence it is of great significance in breeding program. Means new varieties can be developed with help of crossing over. Next one is it provides a physical proof for linear arrangement of genes on the chromosome. Next significance is gene mapping on chromosome is made possible by assessing the crossover value. The percentage of crossover is function of distance between two gene loci. And the last significance is crossing over provides a new definition for the gene. According to this, gene is a smallest section of chromosome with no crossover can take place in between gene, but in between two gene it is possible. The linkage and crossing over are the two different concepts. These are the first, linkage ensures that genes stay in chromosome to inherit together. All the genes inherit together, but in crossing over, this enables the separation of genes in chromosome and separate it into various gametes. Second difference is the strength of genetic linkage between two genes is brought to inversely proportional, is thought to be inversely proportional to distance between them in the chromosome. And in crossing over, the probability of two genes crossing over is directly proportional to distance between them in the chromosome. The third difference is the strength of linkage between two genes can increase if distance between them decreases. Opposite to that, the probability of two genes crossing over decreases if distance between them decreases. Linkage ensures the continuity of parental trait, and crossing over causes alteration in parental traits in passing into offsprings. And last one is linkage reduces the probability of variation through sexual reproduction. But crossing over increases the probability of introducing variability in sexual reproduction. This is about the linkage and crossing over.